And as you know, this is the session on the topic number 2, the immunogenicity topic. I would like just to introduce with a few slides some key points. The topic, first of all, the topic presenter is Daniel Kramer, who is one of the topic writers. He will describe the topic mainly as it has been also written in the third call, a document that you found on the internet and also in your documents here. And however, it should be clarified, as Michelle said this morning in the session, that the work packages that are proposed, I mean the organization, the structure of the work that will be proposed by the presenter and also in the document is indicative, and this leaves room to flexible another approach. Innovative approaches are very welcome, and so you shouldn't feel that you have to stick to the work packages as they are presented in the document. However, you should keep in mind that all the research objectives, as they are defined in the topic, they have to be all addressed. So just the structure can be modified, or according to what you are proposing. Another information is that the EFPIA contribution is in kind, and it is indicate the one that is in the topic and also in the presentation today is indicative in the sense that the EFPIA contribution will be much better defined and fine-tuned, as we heard this morning, during the preparation of the full project proposal, that is the second stage. Because of course that the in-kind contribution will be adapted to the project that will be presented, will be submitted. Then the IMI contribution, as you might have seen in the document, there is an indicative range of 5 to 20 millions for the projects such as the immunogenicity, the project related to safety and efficacy in this call, and the indicative duration is 5 years. Some other things I would like to point out for you and for the preparation of the expression of interest is that you should read carefully the IMI guidelines. Indeed, the IMI guidelines are different from the FP7 guidelines for preparation of projects, and there are a number of differences there, so please take this into account. Then the structure of the expression of interest template that is in the guidelines should be followed, and the expression of interest has to be complete. There are 4 sections. They have to be all complete because the completeness of the document is a criterion of eligibility of your project. So if one section is missing, the project is not eligible. And of course, the expression of interest, as you might have seen, is composed of an A and a B part, and both parts, of course, have to be submitted. Again, all the scientific objectives have to be addressed. The evaluators will carefully look at that. And finally, just a practical advice, because the finalization, once you submit it, you have to remember to press the finalize button there, because otherwise your project cannot be submitted in the electronic tool. But you will find that also in the electronic tool. But unfortunately, there were cases of lack of finalization which penalized then the project. And then I would like to remind you that the rules for participation and submission are on our website. Of course, you have to look at the rules related to the third call. The IMI, you may contact IMI at our info desk should you have any doubt or questions regarding the rules or even the content of the topic. And today, as you heard this morning, there are info booths where people are working with IP and also working with financial part guidelines are available at the entrance there just to answer your questions, and they will be there all the afternoon. Finally, the tool for searching for a partner, very often we have been asked by people who want to either propose themselves as partners or they want to search for a partner. We will have very soon on our website the tool for both purposes, offering your partnership and searching for it. I would like to remind you also that there is a webinar on this on the immunogenicity topic that will be on the 28th of October, and that will be at 1 o'clock. And during this webinar, there will be again the topic presentation by Daniel Kramer, and you may ask questions through a chat box. If you could also probably advise colleagues who couldn't come here to follow the presentation again through this webinar. The last thing I would like to say is that this session, as well as the others, is video recorded. So please, when you ask questions, just speak very clearly in the microphone. Thank you. And then I would like to stop with the slide where I summarize the important contacts. Thank you. So now I will introduce Daniel Kramer, who is from Merck Serono, and his position is corporate expert in immunogenicity, and he will present the topic and will be ready to answer all your questions related to the content of that topic. Thank you.
Thanks. I have to switch that on. No problem. Here we go, presentation mode. Not used to this new Windows thing. Okay. I have it. Yeah. Okay, good afternoon. Thanks for the kind introduction. I tried to guide you through our proposal for the call for immunogenicity. I mean, the exact title is, as you read here, Assessing the Clinical Relevance and Risk Minimization of Antibodies to Biopharmaceuticals. And actually, I mean, as you see here, the original coordinator of the project right now is Kathleen Beach. She's from GSK, but she was not able to come here today. And I'm acting as a topic writer for the topic, but I was in close contact, of course, with Kathleen, and so I will do the talk for her. Okay. So let me start with the need for the need that we see for the public-private collaboration. So, as you might know, I mean, during the last years, the proportion of biopharmaceuticals amongst all pharmaceuticals increased, I would say, really significantly. And in the meantime, it is also known that, in principle, all of these biopharmaceuticals are potentially immunogenic. And essentially, it has really become a major hurdle of developing a biopharmaceutical drug that you have to deal with the immunogenicity problem, which you don't have to if you develop small molecules. And also, the hurdles from the authorities increased over the last years dramatically. There are new guidelines that you have to do, how the assays need to be built up. So the whole thing is becoming more and more important. And interestingly, I mean, although it's so important, we don't really know much about the clinical relevance of many of the detected anti-drug antibodies. So we call them ADAs. So that is the shortcut for anti-drug antibodies. So it's really poorly understood, and also, of course, highly variable because, I mean, there's a lot of difference between the response that one patient is doing to a drug to the other one. And the other thing is really, I mean, at the moment, it's really, really hard to predict immunogenicity. There was a lot of efforts in the companies, so in the EFPIA companies, and also in small enterprises or medium enterprises, and also in academia, to predict immunogenicity, because that would help us, first of all, to develop less immunogenic drugs, and also to already foresee what could happen. And although there was a lot of effort in there, up to now, it's really not doable. And there are several reasons for that. And one is, of course, because nature is complicated, as we all know, and I think our immune system is one of the most complicated things within the human body. And honestly, we know much, and we gain, of course, more and more knowledge, but essentially we don't really know exactly why proteins are that therapeutic proteins are that immunogenic. There are, of course, several theories, but still, I mean, there is still some gray zones where we don't really know why. And then, of course, also, I mean, if we don't really know the exact molecular mechanisms, how can we do prediction if we don't really know exactly the mechanisms behind? The other thing is, of course, not every biopharmaceuticals, or not all biopharmaceuticals are identical. I mean, there are monoclonals, there are cytokines, there are growth factors, there are even small peptides where one can discuss if this is a biopharmaceutical or not. So, I mean, you know, the thing that might be true for the one might not be true for the other ones. And the other thing, and this brings me to the real topic, so why do we need a collaboration, is for the time being, the prediction algorithms that many companies developed and that also some academic people developed and also some small enterprises developed, they were just used on single products. Because, for example, in the companies, of course, you just use your prediction tool that you're having to your own compounds. I mean, you don't use them for compounds from another company. So there is not a lot of exchange on these kind of things. And it might be that we're already having some prediction tools or combination of prediction tools that would allow us to do at least for a 70% prediction or something, but simply the data are not shared up to now. And this is one of the things why we think that a cooperation would really be helpful. Yeah, and as I said, I mean, a combination of the research capacities of pharmaceutical companies and academia or small and medium enterprises would allow to compare all predictive tools, because there are a lot of them, interestingly, in the public domain or from academia even. And that would allow us really on a set of marketed compounds, I will come to that, to really draw a conclusion. Is there something behind that? So is a certain combination of some predictive tools really predictive for predicting the immunogenicity in humans, or is there nothing behind? In addition, it's not just about the prediction. I mean, there's also a lot of research done on immunogenicity in the public domain, meaning, for example, patient registries where they really do independent from the companies immunogenicity research on marketed compounds. And also these data are never really brought together. And so we really rely, for the time being in the companies, we really rely on the data that we create in our own companies. And we don't really have exchange of, at least not to that level, on data that are created in the public domain on immunogenicity. And that would really also gain the statistical power to identify, for example, factors which are relevant for immunogenicity. Because up to now, there's just theories, as I said. I mean, some of them are proven, some of them are not. But we don't have the statistical power to really say, yeah, we are sure this is a factor that is not just true for the one protein, but it's also true in general for being more susceptible to an immune response against the protein. And of course, we would also like to share scientific expertise because, and this also brings me to one thing that was said this morning, we also would like to have a little bit out-of-the-box thinking because talking about immunogenicity is also a lot about immunology. So there might be some people who are not really in the field of immunogenicity, but they have a rather sound background in immunology. And probably these people might think of something that could help us and also the small and medium enterprises in developing these biopharmaceuticals. Because, I mean, if you have a sound background in immunology, you might think of things that we in the companies never thought about that. So it's not just for people that already are working in the field of immunogenicity, but helping us understanding the real molecular basis on immunogenicity would also involve immunologists, for example, from universities. So that brings me to the objectives of the full project. As said at the beginning, so one of the major objectives is to investigate the clinical relevance of biopharmaceutical associated immunogenicity. I mean, we also have to be clear here. I mean, we know it for some, of course. For neutralizing antibodies against an endogenous counterpart, we are quite sure most of the time what these are doing. I mean, and that this is critical goes without saying. But I mean, what we are more concerned, or not concerned, but what we would like to evaluate is really, if you have just binding antibodies, not neutralizing, or antibodies that are low titer, for example, or antibodies that are just transient.